Unmuted. Okay, sorry about that. I was just automatically automatically muted, but we're good now.
Okay.
So yes, thanks for the invitation to come and speak to you. Beekeeper associations are one of my favorite crowds because they're already so enthusiastic about the kind of work that I'm talking about, and hopefully this work that I'm going to talk to you about tonight will be of interest as well. And as Fiona picked up on, I still don't get very tired of the fact that I can use the words sex and drugs in the titles of my talks and get away with it. So here we go: sex, drugs and ecosystem services, the paradox of plant toxins in nectar. I'm going to try and, here we go. So before I start, I'm going to get a little bit of something off my chest. We all love biology or natural history, and we've got some great champions of natural history like David Attenborough, the great egg fan, and Kate Humble, the chicken enthusiast. It's hard to know what really came first, our love of biology or their enthusiasm for it on our tellies, but we certainly give biology the thumbs up. And even these days, physics is cool. We've got Jim Al-Khalili, who's enthusiastic about gravity and other aspects of physics, and Brian Cox, of course, the physicist and geeky heartthrob, who's so amazing rainbows seem to come out of his ears. But when our TVs depict chemists, it's a really different story. We're always depicted as kind of maniacs or somewhat dangerous. I don't know whether any of you have seen the series Breaking Bad on American TV, came over to the UK. Mr. Walter White, the errant chemistry teacher who ended up synthesizing crystal meth in a caravan in the desert, or Peter Wuthers, the intellect or academic from Cambridge University, who was given a chance to tell the world about chemistry at the Royal Institution lectures and made it look more like Hogwarts. So I'm not entirely happy about this, but the truth is chemists have got a few sort of skeletons in the cupboard. Obviously, DDT has a terrible history, and it's been followed by quite a lot of other bad chemistries in agriculture, and of course, various other uses of chemicals in other nasty ways. But I argue that actually biology and physics are much more dangerous than chemistry. You're much more likely to be laid up with a biological organism, a disease, or some other kind of illness, or by physics having fallen over. And actually chemistry is much more likely to cure. So today what I'd like to do is to champion chemistry as a subject, give it a positive spin, and hope that you can go away feeling that chemistry is not an impenetrable subject, and one that's actually quite interesting and exciting, and can actually answer and address some of our important ecological questions. So today's talk is going to be about chemistry, and specifically about floral chemistry. I'm particularly interested in nectar and pollen chemistry. And I'm going to be asking whether these compounds in flowers and chemicals have ecological functions. Do they actually have a role in mediating pollination services? And can we actually use this chemistry to potentially provide solutions for challenges that we face in pollination? But I can assure you that there will be some sex and there will be some drugs. I'm going to give myself a laser pointer so I can point around here. So there will be some sex and there will be some drugs. Here's the sex, here's the drugs. This is a cannonball tree with a honeybee visiting it. And I'll be presenting work from my 2 research labs and the work that we've done with our various collaborators. So just to put it into context, as Fiona said at the beginning, I work at the University of Greenwich, and in fact specifically in the Natural Resources Institute. This is the former scientific wing of Britain's overseas aid programme, and so we still have a mission around food security and alleviating poverty in developing countries. We have a big mission in Africa, for example, and we work around agriculture, health and environment, and also the products of agriculture, and try and improve livelihoods and the way some of these institutions operate. More specifically, my own group works in chemical ecology. This is the study of chemicals in nature and how we can use them to understand insect behavior, monitor pests, and perhaps manage them in ways that we call biorational, i.e. ways that don't use synthetic inputs or harm the environment. I've got some examples here. Hoverflies, of course, are very interesting, beneficial insect because they both pollinate, but also their larvae eat pests. Over here, we've got perhaps the most important pest on the planet, Helicoverpa armigera, or the cotton bollworm, actually here eating pigeon pea. and we do work on various other disease vectors and so on. Perhaps the most surprising one is this piece of work here on tsetse flies or Glossina species. These spread sleeping sickness. And our group invented this fake cow. It might not look much like a cow to you, but it looks quite a lot like a cow to a tsetse fly, especially if you make it smell of cow chemicals. And if you impregnate it with a pesticide, when these tsetse fly land thinking they're going to get a bite, they get killed instead. And so this was actually voted in the top 10 university inventions in the UK in the second half of the 20th century. So there you go, some of the areas of work that we have at the Natural Resources Institute at the University of Greenwich. But as Fiona said, I also work at Kew Gardens. Kew is a collections-based institute, but there's a lot of science that goes on, and a lot of that science is associated with the magnificent collections that we have. Most people are familiar with the tens of thousands of living plants that we have in our living collections, and that's where most people see us. But we have one of the largest herbaria in the world with 7 million specimens in it, and more type specimens. These are the specimens that we use to describe species than any other herbarium in the world. We have over 1 billion seeds in the seed bank. We have the largest fungarium in the world. We have one of the largest DNA banks. We have a collection of wood anatomy slides, which are important for protecting the trade of illegal woods, and we have a very interesting economic botany collection. All of these underpin the science that goes on at Kew, often behind the scenes when people come and actually visit. And at Kew, I have a group that has a similar kind of area of interest as the one I lead at the University of Greenwich. It's called the Biological Chemistry Group. We focus on chemicals as well, structural elucidation, compound isolation, and we're very interested in how these mediate biological interactions. And some of those I'm going to be talking to you about today, particularly the ones around pollinator behavior and pollinator health. Now I'm talking to beekeeping associations, so you really are the last people I need to tell you about what pollination is. But I always like this slide because it brings up some of the really important reasons why pollinators are critical. Not only are they obviously of huge value, which is one of the factors that we can use to engage policymakers and politicians to understand the importance of pollinators, but so many of our crops benefit from insect pollination. So they're very important, obviously, for food security and healthy diets. And in fact, they're particularly important for some of the healthiest foods that we eat. All our fruit and veg, many of them will require pollination visits. But beyond food, and this is where most people focus, but beyond food, depending on where you are in the world, and particularly in the tropics, up to 94% of plant species require pollination for their survival, which is extraordinary. So it shows how important pollinators are to just maintaining our natural ecosystems. And of course, the pollinators rely on the flowering plants as their food. But the thing that's always puzzled me, and one thing I've always been really interested in, is why should a pollinator revisit a conspecific flower? If they're all providing pollen and nectar, what would make a bee visit one flower and then go to the same species to make sure that that pollen got transferred and that plant got pollinated? And that isn't actually so obvious. And hopefully some of those ideas will emerge today from the work that we've been doing. Some examples are here. Perhaps the most notable example of specialization is with Morgan's sphinx moth. This is the moth that Darwin predicted would exist when he came across this orchid Angraecum sesquipedale. This was found or discovered in Madagascar in the Victorian era, and 40 years later, another explorer actually found this moth with the tongue that would fit right the way down this amazing nectar spur. And of course, what this means is if the nectar is down the bottom here, the only creatures that can actually get to that nectar are moths with these huge tongues. And so this secures this food source for this pollinator and ensures that this pollinator will keep focusing on this flower and therefore more likely transfer pollen from one flower to another. And this kind of architecture is very common in pollinating systems. This is a photo I took in South Africa in the Botanic Garden in Pretoria, and you can see how the lovely kind of anthers here tickling the hairy chin of this sphinx moth, just the right architecture and shape and size down here again with this ruby-throated hummingbird on Cs alpinea. So these are ways that plants adapt and modify their structures to ensure they get pollinated. But we get chemicals that do the same thing. Delphinidin is an anthocyanin, which is present in delphiniums, and actually pollinators, particularly bees, have an intrinsic attraction, blue color, which is quite fascinating. Actually use color to indicate when they've been pollinated. So when they're ready to be pollinated, they're white. They change color to red when they've been pollinated. And even chemicals that impersonate sex pheromones in insects are present. So here we have a hammer orchid from Australia, and this is the thinnid wasp that arrives, thinks it's going to mate with a female because this thing smells of this chemical. And so it's an amazing way that this plant has adapted to attract a very specific insect to come and visit just its flowers. In fact, it's so specific, things can get really out of hand on occasion. So here's another little bit of sex and drugs for you. But when most people think of pollinators, they think of, well, this association's presumably best friend in the animal kingdom, the honeybee, Apis mellifera. This is because it's such an engaging creature. Most of us know something about it. We've been brought up, and particularly in the last 10 or 20 years, the stories about bees have become much more positive than they were perhaps in the 60s and 70s when bee movies like The Killer Bees were around. But of course, honeybees are not just Apis mellifera. There are about 7 different species of honeybees around the world, most of them coming from Southeast and South Asia. Perhaps the other most important one after Apis mellifera is Apis cerana. This is cultivated or kept as effectively livestock across East Asia and down into India too. Although the largest of the honeybees, Apis dorsata, which is the one that produces these beautiful, magnificent combs off buildings and trees, is actually harvested by some of these daredevil collectors in parts of South India and Nepal. So quite a diverse group, but of course, 7 species is nothing. There are 20,000 species of bees estimated across the globe, and they're all magnificent, and they all have a place in our hearts when we're thinking of bee conservation. And that's one of the areas I always like to champion when I'm talking publicly. We've got here Bombus terrestris, a bee that's probably familiar to most of you, on a rhododendron flower. I'll come back to rhododendron later. Again, I'll come back to this species here, Andrena scotica, which is a mining bee species, this beautiful cuckoo bee from northern Australia. And here we have Caletes hederi, the ivy bee, which I'll also come back to again. So bees, very diverse, all very important. We're trying to get this message across by the use of the hive at Kew. Some of you may have been to see this. It's a very interesting structure, very compelling and captivating. Doesn't look much like a hive. I think most of you will agree, although it's made of lots of hexagons. That's about as close as it gets, and it's sort of box shaped. But it's been a great asset for Kew to draw attention to the reason why conservation and the conservation of plants is so important. It was originally built to draw attention to the plight of honeybees, but actually we've turned it into a tool to talk much more about bee diversity. And in fact, my postdoc, who will feature later in the talk, Hauke Koch, a German bee specialist, in the last 3 years has collected 107 species of bees just in Kew Gardens, just to show how diverse bees are and can be in urban settings like Kew. But it's a great asset for us to champion the plight of bees. But of course, it's not all about bees either. There are somewhere in excess of 100,000 species of invertebrate pollinators in total, and these include beetles and flies, hoverflies, and butterflies, of course, and even many vertebrates, pollinators, and some of them are actually economically quite important. And it's this diversity that I think is really, really fascinating. But as I say, also very important for maintaining natural ecosystems and also producing food. So for instance, if you look at this pie chart here, you can see that honeybees are probably still the most important because of course they're used commercially to produce a lot of foods, but wild bee species probably comprise about 50% of all the pollination services required for food production in the world, so of the plight of wild bee species. But at least 25% of the rest of the pollination service is provided by species other than bees. And so we always must bear all of these species in mind. Perhaps the most surprising for many is this bat, which is the main pollinator for durian fruit, a rather smelly Southeast Asian fruit. Smells a bit weird, tastes lovely. And I'm sure many of you know that this tiny creature here is a biting midge, Serrata pagonid, which is the pollinator for cocoa. And you can see how tiny this creature is compared to the flowers. This is a dime here. And this is the flower, of course, that produces cocoa pods from which we get chocolate. So we can thank a biting midge for all our chocolate. There is, of course, one other vertebrate pollinator in the world, and that's us. We're required to do pollination service where there are no pollinators left. In parts of East Asia, it's actually fairly common to see people actually doing the pollination service in fruit trees. In Australia, buzz pollination is important in tomatoes, but in Australia they don't have any of these beautiful bumblebees, so they have to do it themselves by hand. But of course, this is a rather serious indictment of the fact that we're not able to look after our natural ecosystems to the extent that we can maintain essential services from pollinators. And this has been highlighted fairly recently in the IPBES Pollinators, Pollination and Food Production report by Simon Potts from Reading University and other colleagues around the world. Thousands and thousands of papers were read for this and put together. It's an absolutely astonishing document if you can get your hands on it. It's a fascinating read and still highly relevant now, despite already being a few years old. But perhaps the most shocking thing is that actually, when we talk about the plight of pollinators, there's certainly evidence for some being endangered, some being critical and vulnerable, and others being near threatened. But actually, for most species, we're deficient in data. We simply don't know what their status is. So there's a huge amount of work that needs to be done to really find out where we are with our pollinators. And of course, they're exposed to many stressors that could cause some of these potential declines: climate, habitat loss, land use change, perhaps one of the most serious. Pesticides have probably had more attention in the press than any other challenge, and also parasites and diseases. But my interest is around this, the floral chemistry, and how this can help us understand or influence any of these other challenges, and potentially have direct negative or positive impacts on the bees themselves. And that's what I'd like to focus on for the rest of the talk. So I've studied chemicals in plants most of my working life. Actually, I came to pollinators a little bit late, and before I was working with pollinators, I was actually mostly interested in the fact that plant secondary metabolites, these strange chemicals, wonderful array of different chemicals that plants produce, were there for actually defense. Of course, plants can't move. They have to defend themselves where they stand or are rooted. So they invent fantastic ways to defend themselves. Most of us have experienced a sting from a stinging nettle, or maybe even a prick from a cactus, but actually most defense is chemical in plants against diseases and against herbivores. And these chemicals can be really quite toxic. But what we've found increasingly in the last few years of our research is these compounds, the same compounds that are there for protecting the plant against insect herbivores, these antagonists, also turn up in the nectar and pollen, where they are interacting with mutualists. So it's a paradox that we have these insect toxins showing up in nectar and pollen. The compounds I've got here, this is granatoxin, a compound I'll talk about in a minute, and this compound again will feature later. This is caffeine, again another chemical that is produced by plants as a defense. So I covered this in a review in 2017, and I've just revised this now in a review that's literally just been published today, and I've shared that with Fiona. And if anybody would like to read up more on the topic I'm talking about today, do please ask Fiona for a copy of this paper. It's open access and freely available. But in this paper, I talk about these different ways that we think these chemicals might be interacting with mutualists. And the first is mediating pollinator specialization. You saw some examples of that earlier. The other is antimicrobial activity, potentially as bee medicines. plants produce chemicals to protect themselves against diseases. So is it possible that these chemicals could have a similar effect and protect bees from these parasites as well? Protection against nectar robbery. Nectar robbery is a strange phenomenon in plant pollinator interactions, which I'll talk about. And then potentially, can plants actually drug bees to be better pollinators, which is perhaps my most interesting subject. So I'll leave that one to last if you wouldn't mind. But the one I'll kick off with is this fifth one, plant-wide biosynthesis of plant defense and unregulated, and what the consequences of that can be. As I say, mostly we assume these chemicals are produced in the plant as a defense mechanism. So plant-wide biosynthesis of defense chemicals. The example I'm going to use is the one of lupins. Many of you might have lupins in your gardens, and you might be familiar with this kind of stalk where you've got loads of aphids on the colony. Aphid colonies rather on the stem, and this is because these are highly susceptible to aphids. They've actually been bred to be low in this toxin called lupinine, this one here. In Peru and Ecuador, they actually grow a species of lupin called Lupinus mutabilis. It's an important food, but they haven't bred out this toxin. It's actually quite high levels in all parts of the plant, and even in the beans that they then eat. And what they do is they put them in a sack, hang it in a river for about 3 or 4 days, and all the toxin gets washed out by the river. Then they bring it up, then they can use it for food. And I was fascinated to know, well, if this is a pollinator dependent crop, which it is, what consequences might there be for pollinators if they interact with pollen that contains this toxin in the when they're pollinating? So we undertook some studies with Bombus terrestris colonies. I was hoping to be able to show you a video here. I might need to, that's why I have to use the regular pointer. And you can see here's one of our colonies. Actually, bumblebees are quite easy to rear. We can either collect them from the wild. I'm going to turn my sound off so you don't have to hear the commentary in the background. So we take one of these colonies and we provide them or provision them with pollen that contains this compound, which we've isolated from other parts of the plant. And we tested it to see if there was this spillover effect on pollinators. And we found that at concentrations that we find in the pollen, which is between 1 and 2 milligrams per gram of pollen, we actually do get fewer offspring, many fewer offspring produced. So there isn't a lethal effect, but there seems to be some colony effect. They're producing fewer offspring. And actually, if you measure their thorax width, which is a very good indicator of their size and their development, we actually get far smaller and significantly smaller progeny from these colonies that are fed this toxin. So the likelihood is that where farmers are actually encouraging the production of this compound in their plants because it protects them naturally from these pests, there is this spillover for the bees, the pollinator. And this could actually have quite serious consequences if the landscape is dominated, as it would be, in an agricultural setting. This is a photograph of some of the higher grounds of Ecuador, where Lupinus mutabilis grows quite commonly. So there could be potential interactive effects, for instance, with other stressors, for instance, pesticides. So maybe when we think of host plant resistors as a great alternative to pesticides, we need to understand fully the consequences of these processes. So the next example is going to be one on nectar toxins and how they might mediate this phenomenon of specialization in pollinators. But before I do that, I wanted to give you an illustration of how we know some of these compounds are really there for defense and not there actually for the pollinators. This is a bit of work that was undertaken by my colleague and myself, Alison Scott Brown here, looking at this chemical called guanotoxin and looking to see how toxic it was to insects and whether it could be the reason why the young leaves here you can see on this sprig of rhododendron seem to be protected, whereas the older leaves tend to be very susceptible to this greenhouse thrips. This is the compound here. You don't need to be a chemist just to know that it's a structure. It's soluble, which you find in the nectar. But we looked at the concentration of this compound in the leaves at different ages, and we find the highest concentrations, this is the increasing concentration along here, were always in the youngest leaves. These are the oldest leaves up here. And we did some bioassays that showed that at concentrations we find this compound in the leaves. It was potent enough to kill all of these thrips, so naturally protecting these young leaves, prioritizing this new growth over the old growth, apologies. And so we have this chemical that's produced by rhododendron as a defense against herbivores. Now we know that this chemical also occurs in the nectar of rhododendron because we know that rhododendron honey produces this compound. And in fact, it's been known for thousands of years. In fact, Mithridates VI of Pontus, who claimed to be the archenemy of Rome and is possibly the grandfather of plant chemistry, also knew about this toxin in rhododendron. And when he was fleeing the invading army of Pompey the Great in 66 BC, he left loads of honey on the path. And so as the troops of Pompey followed on, chasing after Mithridates, they encountered these honeycombs and assumed they were gifts of the gods, started to consume them, and then were poisoned by their toxins because they're effectively a sodium channel blocker, effectively having the same mechanism on the body as many synthetic pesticides, and it causes impaired consciousness and potentially arrhythmia of the heart, and potentially even heart attacks in very rare cases. But in this particular case, Mithridates knew that if Pompey the Great's army would eat it, then he could go back about an hour or two later and find this army in a stupor and slaughter them, which is in fact what he did. And Pompey lost about 1,000 troops. So arguably the first example of chemical weapons of war. But the reason we were interested in this was because we knew that nectar in this plant, Rhododendron ponticum, could potentially be harming bees in the British Isles because it's a hugely important invasive weed. And in some parts of Ireland, it's the only plant around. This is a hillside in Southern Ireland, in Kerry. It's an invasive plant in the British Isles, but it's certainly found its preferred home. Its native distribution is here around the Black Sea, with a couple of isolated spots in Spain. The likelihood is that these are remnants from the Ice Age, and now actually the UK has the perfect environment for this plant. That's why it's doing so well in the British Isles. And this is work we did with Jane Stout at Trinity College Dublin. So we were interested to know about the chemicals in the nectar, this granotoxin, this insect toxin, and what it would do to bees. So we undertook some bioassays with honeybees and with bumblebees, and you can see in this figure here that when we feed this compound, granotoxin, this chemical that we find in the nectar of granotoxin, to honeybees, they die within just a few hours. And this is them being fed artificial nectar plus the toxin at natural concentrations. This red line is them just being fed the nectar. And we can't do this red line with bombus because, one, they're completely unaffected by granotoxin, and secondly, they eat a huge amount, so we can never collect enough nectar to feed bumblebees. Honeybees die within 6 hours. Bumblebee species, we can feed them for days and days and days, and they're completely unaffected by this compound. So they seem to be adapted to it. They're unaffected by it, which is a really fascinating thing because these are actually quite closely related species. We also were very lucky to find an Andrena scotica collection of nests near to the department, and so we were able to work on Andrena scotica too. We tested it against Bombus terrestris. We weren't able to do mortality tests because Andrena scotica wasn't as conducive to staying alive in the lab as Apis mellifera and Bombus species were, but we did some work on flying time. With Bombus terrestris, there's no effect. With Andrena scotica, they spend virtually no time flying when they've been feeding on this compound at natural concentrations, and they spend more time showing symptomatic behaviors, grooming and paralysis. So they might as well be dead because they're not foraging for their offspring. So there's a huge environmental pressure to clear rhododendron, and also this idea that it's now toxic to honeybees has not helped. But the truth is, rhododendrons produce perhaps the 10th largest amount of nectar per flower than any other plant in the UK. And they're very popular with bumblebees. So we have a bit of a conservation conundrum here. Do we get rid of all the rhododendrons, or do we consider it an important source of nectar for bumblebee species? The reason why we think that there is this adaptation from bumblebees, they've adapted because they have been selected by the plant as the preferred pollinator. We've actually looked for this chemical, the same toxin, across the whole group of rhododendrons. And in rhododendrons, there are about 1,200 species. We've looked at about 200 of them, and these include many with very interesting specialist pollinator syndromes. These two at the top, for example, this is a bird-pollinated species of rhododendron called Rhododendron mellifagidum. It's bird-pollinated by a bird a little bit like this honey eater here. In fact, this honey eater is restricted entirely to flowers of Rhododendron culminicola. So this is a fairly interesting phenomenon in rhododendrons, particularly down in kind of Malaysia and New Guinea and some of the southern Southeast Asian islands. This is specialized for long-tongued moths. And as we go down to the generalists, you get these more open pollinated ones, which basically are accessible by any kind of all-comers. And what we find is that where the plants have specialized anatomically or physically, they show no signs of the toxins. Whereas when they're a generalist flower, so open to all comers, they nearly always contain a toxin. And this association, we think, is evidence that the plants are either producing a physical change to moderate or mediate the visitors that they get to encourage specialization, because this guarantees or more likely guarantees pollen transfer between conspecifics. Whereas down the bottom, they're using this toxin as a way of filtering out non-adapted species. So the third example are nectotoxins where they moderate parasite load in bees. And this is building on the idea that the possibility that plant toxins that are produced to protect a plant from diseases of plants could actually have similar effects on bees. And this is a graphic that we used in a recent paper with my postdoc, who I've already mentioned, and my colleague Mark Brown at Royal Holloway, looking at antimicrobial effects of nectar chemicals. When bees visit flowers, this is a place where they transmit diseases. It's very well established now. And in fact, there's more work showing that there's interspecies transmission of some diseases now. And these diseases modify foraging behavior. They make bees less able to forage effectively, and they have other consequences, of course. But when bees visit these flowers, they do encounter these interesting, potentially interesting antimicrobials produced, we presume, for the plant, but we consider the possibility that they might be able to kill pathogens of bees or moderate transmission when bees are interacting at the same site, and potentially inhibit proliferation when they've been consumed by the bee. And so Hauke then set about in my lab at Kew Gardens to find out whether we could find any evidence for this. Now, if you've ever collected nectar, you'll know that plants don't actually produce very much, and bees do us a huge favor by collecting all the honey that they do, because this saves us doing the job, especially the monofloral honeys. And we took a whole bunch of monofloral honeys and we tested them against this pathogen of bumblebees. This is a pathogen called Crithidia bombi. You can see it wriggling around here on the screen. I think this is actually one of the only bits of footage of this pathogen. It's debilitating for Bombus terrestris. You can see Bombus terrestris here actually feeding on ling heather, which is this heather. We found this heather to be the most biologically active at controlling this pathogen. And so this was really fascinating. So we thought we'd better look at this in more detail. Actually, Echium vulgari honey was also very effective. And actually, Arbutus unido was also quite effective. This is the strawberry flower tree, and even Tilia species. Now, this is the lime tree or linden. Most of the others were not significantly different from the controls here. So we've got this particular target now, ling heather. When we test it in isolation, in vitro, not in the animal itself, we find it's biologically active against this gut parasite of bees. So what we did was we used some fancy equipment in our laboratory to isolate various compounds from the heather, and one of them that we isolated was this compound which we've called calanine. And we tested this at various concentrations against the same pathogen, Crithidia bombi, at different concentrations. And you can see in the control, so no chemical, we have a count of about 15 cells per nanoliter of the pathogen. And if you go up to about 10 parts per million, we have no change. But as you increase the concentration up to what we find in the crop of bees feeding on Calluna flowers, we find that we get absolute inhibition of this pathogen. So it looks like this chemical, which occurs in the nectar of the flowers and in the honey of Calluna heather, is actually controlling this pathogen for the bee. And in this slide, I'd like to show you what we think to be the mechanism. We treated different Crithidia bombi cells here, and we show that when you treat them at this concentration, when they're exposed, almost all of them end up without a flagellum, this little wiggly tail that you saw, whereas in the untreateds, they all, nearly all, have a tail. Now, this is really important because if I can show you this video here, which will trundle along, this is the Bombus terrestris ileum, the gut, the hind gut, where Crithidia bombi resides when it's infecting the host. And you can see all this wriggling around. These are all the pathogens embedded into the gut wall. Now, if I pull this along a little bit more and I show you a close-up, you can see here's a pathogen here. This is the body, and that's the tail going into the epithelial cell wall of the ileum. It uses the tail not only to swim around, but to anchor in the cell wall of the ileum, where it then proliferates, produces more, and consumes food in the bee. Without its tail, the activity of this calanine, it's no longer able to do this, so it gets flushed out and is never able to establish an infection. So we have a chemical here produced by a widely growing plant in our natural ecosystems that bees can consume to control their diseases. But we are, of course, facing challenges maintaining some of our natural habitats. This is an example of heather blossoms on Long Mynd in August 2017, and this is the same site in August 2019. About 75% loss of the flowering on this slope, potentially due to a pest that's arrived, but also due to climate change. So we really need to understand much more about how our natural ecosystems are supporting our pollinator populations and these important interactions of our ecosystems. And this is a really good example of how we need to put pressure on to maintain these natural processes. I'm going to quickly skip past nectar toxins because I realize that I'm getting a little bit oh no, I can't resist this one actually. If you just forgive me, I'll whiz through it. Aconitum nectaris are specialized for long-term bumblebee species. Some of you will know this flower. This is the hood, the galea of Aconitum. The name escapes me at the moment. This is Aconitum lycoctonum down the bottom at any rate. These are two Aconitum species we have growing in the gardens at Kew. They have these kind of strange nectaries underneath this hood. And these are a specialized position for long-tongued bumblebees. You can see one coming in here in this graphic, and it's able to reach those nectaries. But short-tongued bumblebees like Bombus terrestris aren't able to. In fact, this species of Bombus gerstaceri feeds exclusively on Aconitum, and you can see it's got quite a fantastically long tongue there. But these short-tongued bumblebees are pretty clever. If they can't get to the nectar through the normal route, they will chew away a hole in the top. And you can see a hole here where they've chewed through to get to the nectar. And I was wondering whether this was a real problem for the plants, whether there was a fitness cost. So we undertook a survey in the gardens at Kew. Most people rely on an MSc student to sit in the baking hot sun with a notepad monitoring bees. We did do this actually, but we also compared it to this fascinating device developed by my postdoc Sarah Barlow here, where she had motion activated cameras that only monitored visits to these flowers to try and figure out which bee species were doing what. And you can see we've done this on both the Aconitum species, Aconitum lycoctonum and Aconitum napellus. This is data we've got for Bombus hortorum, the actual pollinating species, the long-tongued one. You can see we get up to 6 visits an hour on Aconitum lycoctonum, maybe 2 visits an hour on Aconitum napellus to each flower. And the number of visits we get is nearly 1,000 by the pollinator on lycoctonum, nearly 200 on napellus. In the same time period, we only got 20 visits by Bombus terrestris, the robber, and only 3 of those were actually robbing events. So these robbing events are actually very rare, and we wanted to find out why they were so rare. And in fact, in Bombus terrestris on Aconitum napellus, we found virtually no robbing events, even though we'd seen evidence of it. We were never able to record it. So these are very rare events, and so probably not actually too much of a consequence. It turns out that these Aconitum species produce a toxin called aconitine and various other aconitine alkaloids in the nectar. These are actually deterrent to the nectar robbing species, but also the pollinating bombus. It's just that the pollinating bombus have developed a much higher tolerance to these. So the plants have a sort of trade-off between deterring robbers and attracting pollinators. And this is mediated by fluctuations in the concentrations of this toxin in the nectar. But ultimately what this means is the plants don't suffer unduly from a robbery in the field. So the final example I'd like to give you with respect to how plants in nectar affect bees is perhaps my favorite example, and this is the one where we think we can justifiably say that nectar toxins manipulate pollinator behaviors to the point where the plants are actually drugging bees. Now, caffeine, as I mentioned very much earlier, is a defense chemical. Plants produce it, for instance, in the young tea leaves or in beans of coffee to protect them against herbivores. And actually, they're distasteful to bees as well. But we found this compound also occurring in the nectar of coffee and citrus flowers, but always below the bees' taste detection threshold. So the bees can feed quite happily on nectar that contains caffeine because it's below the threshold that they have to taste it. Just as a point of reference, this is how much caffeine you would get in a cup of Nescafe. So it's actually quite high concentration still, much higher in some of the coffee species than in the citrus, but we were fascinated to find out why this was. Now, to understand our thinking on this, you need to understand that memory, and many of you will know this, that memory is a hugely important part of successful foraging, particularly in generalist pollinators, learning and memory. This is associating good food reward with certain cues like colors or shapes and flower odors. This is very important for successful foraging in generalists. And we wondered whether caffeine could be inhibiting potentially the memory formation or even embellishing it. And so we use this assay called a proboscis extension reflex or response. That's one of the specialist tools of my colleague here, Jerry Wright, now at Oxford University. And you can see in this video clip, this is a tethered bee. No bees were harmed in the making of this movie, I can assure you. This is a tethered bee being fed nectar. And as it's being fed nectar, an odor plume is being blown across its antenna. So you do this about 6 times with a 30-second gap in the middle. It very quickly learns to associate that smell with good food. Now, in this second video from Rudolf Menzel in Germany, you can see a similar tethered insect. Sorry, I'm going to stop that. You can see a similarly tethered insect, and this one isn't going to be given any food, but it is going to be given the odor. And when the red square comes, that's when the odor comes. Out comes the tongue because the bee thinks it's going to be given food. So it's remembered that smell. So this is evidence that bees have memory. And we can use this to actually measure how effective bees are at making memories. One of the main reasons that Jerry has used this actually is to look at how pesticides influence memory formation. And as you might expect, it's negatively. But amazingly with caffeine, it actually enhances the memory of odor cues associated with food and at the concentrations that we find in the nectar. In this graph here, you can see this, we've got test time after conditioning. We've got after 10 minutes, we just give, we've got sucrose fed bees or sucrose plus caffeine fed bees. And we can't really find a difference in those that are able to remember the learned smell, although the caffeine ones are a little bit better at it. But after 24 hours, most of the ones that were trained without caffeine have forgotten, whereas the ones that were trained with caffeine can still remember that smell, and they still remember it more than untreated after 3 days. So this is quite amazing, and a very clever scientist at Dundee, who was one of our co-authors, Mary Palmer, did some fancy work with bees' brains and showed that actually caffeine depolarized the membrane potential of these amazing Kenyan cells in the bee's brain, where bees actually form these memories of association, where they are learning to associate two stimuli at the same time. And because they're depolarized, they're much more likely to fire. So these bees are being drugged by caffeine produced by the plant to be better able to remember the source of that food, so they're more likely to return to it and pollinate it. So really quite a fascinating bit of biology and nature. Now, what we've tried to do is wonder whether there's any way we can use this to resolve a little bit of a problem that farmers have. Many of you will know that farmers not only these days use honeybees for pollination service, but they also use bumblebees, particularly in the colder, earlier parts of the season, particularly in soft fruits. And one of the consequences of inadequate pollination service in fruit production is misshapen fruits like this strawberry. We wondered whether we could improve the forage service, or rather the pollination service provided by these bumblebees that farmers spend a huge amount of money on. So with my postdoc at NRI, Jan Dudenhofer, we set about a series of experiments where we would prime the bees. So down the bottom here, you can see here's my caffeine molecule, here's sugar, this is the food, this is the drug, and this is the smell. These are the smells of the strawberry flower. So in the hive, what we're trying to do is we're trying to encourage the commercial bees to associate good food with this smell and make them do it very much more effectively because of the caffeine. And this is a weird device that we're putting inside these commercial hives. And then we're letting them either forage in these artificial polytunnels or in these flight arenas in the laboratory, and then monitoring their behavior. And I realize I'm running out of time here, but I will try and complete this part of the talk. In the laboratory assays, what we showed was that primed bees decide more quickly. Here you can see time until first choice. The primed bees make decisions much more quickly, and they waste less time on a single flower. And overall, this results in more visits for foraging time. So caffeine actually could improve the foraging behavior of these bumblebees. And then in our artificial polytunnels, you can see here we've got the bumblebee colony. Here are our strawberries, our strawberry plants. We've got distractor plants at the end, lavender. They love lavender. But actually, when we give them caffeine and odor, we find that the proportion of bees visiting strawberry flowers is significantly higher than with just the odor and with neither. These might seem like very small percentages or differences, but actually when you're a farmer and you're producing food, if this can deliver over 10% increases in yield, then these are really worth having. But amazingly, we also found that these caffeinated bees produce better quality and even heavier fruit. So we think that we can actually use this amazing ecological phenomenon to improve the pollination service of commercial bumblebees for fruit farmers. And we're just actually hoping to get a new project in to look at this with other fruits like raspberries and strawberries and work much more with farmers in real field settings to see if we can demonstrate this at field scale. I realize I'm kind of getting a little bit over time now, but I did want to also mention one other really interesting phenomenon. You'll probably all know about this issue with tilia trees. This is a beautiful tree that we got at Kew, where we find lots of dead bees under it. For many years, people thought it was because the plant produced mannose in the nectar. Mannose is toxic to bees. Turns out it isn't there. Some people have claimed that there is this pesticide that's occurring in there, but of course you still get mass bee deaths under trees that haven't been sprayed or treated. But one thing that we found recently was caffeine in the pollen. And actually, what really happens is you don't get honeybees, you only get bumblebees. And bumblebee workers are really the only workers that spend much time feeding on pollen. They need the protein. And so they're much more likely to be consuming caffeine when they're visiting these flowers and making these strong associations of good food reward. And then they come back and they revisit the flower long after the plants have stopped producing nectar because all the pollination service is done. but they're so convinced that this is a good food source, they keep flying around until they run out of energy and end up like this poor girl lying on the floor. So we're finding that caffeine's popping up in quite a few other places, and maybe not so unique to coffee and tea as we might have previously thought. Sometimes the stories that we tell can make it into the House of Lords, but this is a bit of a worry because they don't often get told with quite the same accuracy. We now have Lord Stevenson of Balmacara, no relation I might add, who's now planning to eradicate all the rhododendron from his garden because he thinks it's poisoning his bees. And the truth is, as I said before, it's actually providing a lot of nectar for a lot of species other than honeybees and one or two of the mining bee species. So we need to be careful how we provide messages to policymakers and those that are listening and those that want to be active and activists for conserving bees. And this is actually very much the case with saving bees too. It's my belief and the belief of many others in my field that sometimes the messaging around bee conservation is oversimplified. If you put in save bees into Google, the pictures and ideas that you get will nearly always be about saving honeybees. And actually, in truth, in most parts of the world, while bees, honeybees fluctuate, there are declines in some places, actually they're not really in decline. These are managed livestock. They're very important, and don't get me wrong, I think they're a fantastic engagement tool for people, but we need to really be starting to think a little bit more widely. And certainly this fashion and trend for keeping bees in cities needs to be reconsidered. London, where I live, I'm living down around this corner of London, somewhere about here, just near Wimbledon. London's about 47% green space, so arguably bee friendly. We've got millions of trees, thousands of wildlife sites and parks, but many of the gardens are being turned to paving as people want to have more convenient living spaces. Much of the land is amenity grassland, so we can't really do very much to change it because it's football pitches or cricket pitches and so on. So the opportunities for creating pollinator forage are limited to perhaps half of what we've got. But that's what we need to be doing. We need to be doing much more to provision for pollinators, especially if we want to continue with our interest in beekeeping. So the question I'm asking is to beekeep or not to beekeep, and that's the question for certainly for UK towns and the bigger cities. According to the London Beekeeping Association, I've been in touch with Richard Glassborough recently, who's the current chair. The number of honeybee colonies in London has more than doubled in 10 years and continues to rise. But probably most hives aren't even registered, and colony densities in some parts of the city are raising concerns. Excuse me. You can see down here in some locations we've got over 50 colonies per square kilometre. Now, in a paper recently published by by Alton and Ratneeks and the biologist, they estimated that one hive needed about 0.83 of a hectare, or nearly a hectare of lavender. Now, this isn't looking much like London to me. This looks much more like London. And this is something that you'll often see. You've got 10 hives here on the top of a commercial roof doing their bit for the environment, a little roof garden producing nothing like what this collection of hives would need. And so all these hives are flying off to various other corners of London trying to find food. And so we've got a real problem on our hands. And we've just done an assessment of this in a new paper I'm going to be publishing later in the year, when we publish Q's book on state of the world's plant and fungi. We've got data from London's green space, from Landsat, and bee colony density from this year from APHA, Animal and Plant Health. And using Alton and Ratneeks' paper, we estimated that in London, it could support about 7.5 colonies per kilometre, or about just over a tenth of a kilometre of forage per colony. So if you look at this map that we've developed here, you can see that the white to the green represents what we consider to be sustainable forage in London, and the purple and pink what we consider to be less than 0.133 kilometres squared of forage per colony, so not sustainable for current bee colonies. And so one wonders what's happening to the other wild bee species that are outcompeted for this limited forage. And also disease transmission, as I mentioned earlier, between species, from honeybees to wild species, is now an established phenomenon. So we really need to start thinking more carefully about beekeeping in cities and what impacts it might be having. It might be that our enthusiasm to save bees is actually having the opposite effect. What we need to do is do more to enhance these landscapes so we can accommodate some of our honeybees, but also maintain some of our wild ones. One of the things I find very interesting is this talk about keeping gardens untidy. Of course, there are limits. We don't really want to see this just over the garden wall, but these people actually got fined in Chatham. And actually, this is mostly weeds and ivy. And of course, ivy is a hugely important forage for this beauty, Colletes hederi. So we also need to be mindful about what we consider to be untidy and unacceptable in terms of our city and town living scapes. But what I would really love to do is to encourage more people to appreciate some of the wild species of bees in our cities. These are all videos taken in my tiny garden in Wimbledon. This is a beautiful Osmia bicornis male, just emerged from my bee nest, looking very happy, about to go off searching for a female. I told you there'd be sex in this talk, and you can see here down the bottom, this is the lucky male, just a few moments later having found themselves a female. Off he goes up here. And in here, I've got some cat's ears that I've allowed to grow in my lawn because I don't mow it very much. I think this is a Lasioglossum feeding at the moment. And in the background, you can see Dasypoda hirtipes, you can see the pantaloons on the bee there as it comes into forage. And then up the top, perhaps my favorite video of all, this is Anthophorum plumipes, my earliest bee in the garden. One of the things I love about this video is the way that the bee cleans its proboscis after it's been feeding. And what a lovely long proboscis that is. That's one of the longest tongues we have in the UK in bees. And that, of course, allows that bee to specialize on some of the longer or deeper nectaries like this pulmonaria flower that it's feeding on. I'll just let you enjoy that moment of tongue cleaning one more time before I finish. Look at that, isn't that amazing? Quite extraordinary. So this is all just wild bee species in my garden in Wimbledon. And I think we need to do much more to encourage people to appreciate pollinator diversity. So I'm not going to recap. I'm just going to go straight to the acknowledgements because I know I've gone slightly over time. I promised Fiona I would try and keep it down to 50 minutes. But I have to acknowledge all my collaborators. None of the work that I presented today would be possible without all these fantastic and really quite brilliant friends and colleagues from various universities and institutes around the world. So thanks very much to them. And thanks to all of you for listening. I'm very happy to answer any questions that you might have.
That's brilliant, Phil. We really enjoyed that. And what you were saying about London forage dovetails very well with it.
I hope you're all still there. I can't hear Fiona or anyone coming back.
Hang on, can you not hear me?
Do I need to give the, can you take back the?
Yeah, maybe you.
Ah, there you are, Fiona. Hello.
Hello. I don't know how.
Do you know how to get me back?
I don't know what's going on.
I'm not quite sure how to send. I'm not sure how quite sure how to send it back to you. I mean, if you wanted, I could. If there was a chat function, I could just simply answer chat questions. Would that be possible?
There is a chat function. And there are a couple of questions. So, a lot of people have said thank you, very engaging.
Trying to look to see if I can find out where to do that. I can see all the people. I'm afraid I have no idea how to send it back to you.
I think if you press on my name, then you can
I can see everybody. I can't hear you actually, Fiona, which is a bit of a problem.
Oh, okay.
Can you wave your hand if you can hear me?
I can hear you.
Ah, okay. So you can hear me, which is good. Right. I've got a chat here. Ah, now I've opened the chat. So Sue Carter says, would be interesting to see if calanine has an effect on nosema. Yes, that's a great question, and it's something that we're actually looking at the moment. So yeah, watch this space. Hopefully we'll come back to you. But we do have a paper in prep that actually has found caffeine in another plant in the UK, a plant called Onobricus, and there's a very specialist bee of Onobricus but I mean, one of the things that we think is really interesting is Onobricus actually also contains caffeine in the nectar, and that caffeine is biologically active against nosema. So there's a lot of work coming out in this space, and perhaps much more that serve relevance to honeybees. So Gerwin asks, does electrical conductivity play a part? Sodium was mentioned earlier. Sodium blocker seems to be key in medicine for message transmission and from the brain. Well, I think these are slightly different mechanisms. One of the things that sodium channel blockers do is stop nerves from turning off when they're firing. What caffeine did was actually reduce the action potential, which is a slightly different thing. It gets it to a point where it's nearly firing.